Hi everyone, thanks for joining. I'll start the class now, maybe in 1 minute. So today we'll be talking about the immune system, and if people have questions, you can ask me about other topics as well. Right. Yeah. Okay. So I hope everyone can hear me and see the screen that I'm sharing. So, right. Today we're going to talk a little about the immune system. You guys learned about some of the important immune organs briefly also during your 12-week course. And I thought I'd go into it in a little more detail about some other organs and recap on what you learned about the lymph nodes as well. Because this system, immune system, is an extremely essential um, component of our bodies, making sure and the immune cells are literally infiltrated throughout the entire body. In every organ you have immune cells, including the brain, that protect the tissues from harm, from pathogens, try to detect cancer, right? So it's a network of biological systems, as you can see here, that protects the organism from diseases. It can detect against a whole host of different kinds of threats, pathogens, viruses, bacteria, parasites, as well as, of course, threats that appear from inside the body, like cancer cells or even foreign objects. If you have like a thorn stuck in your finger, immune cells are what make it hurt. Immune cells are what detect that this is not supposed to be there. And the reason that you have redness and swelling and pus in that region is because there's a war going on, um, orchestrated by your immune cells. And it's very important for the immune cells to do two things, right? One is to detect diseases, detect any infected cells or possible pathogens or cancer cells. So detect things that are harmful, but also to make sure that they don't detect um, self cells. So cells of our body that have done no harm, that are just normal functioning cells, the immune system should not react against them. So making sure that immune cells are detecting the correct thing as a threat and not just reacting against anything and everything they see is also another very important function of the immune system, right? To make sure that there is no, as they call it, autoreactivity. So part of all of this, you can sort of divide up the essential components of the immune system into different types of immune organs, primary lymphoid organs and secondary lymphoid organs. As you know, the lymph fluid is one of the main carriers of a lot of the um, immune cells and lymphocytes and so on. A lot of them travel through the body via the lymphatic network. And some of the organs that contribute to this to the immune system are the bone marrow and the thymus, very important primary lymphoid organs. This is I'll talk about them in more detail, but essentially the essential site of hematopoiesis and T cell training. And another set of organs are the secondary lymphoid organs. These include the lymph nodes, the spleen, the tonsils that you guys must have heard of. Some people have them removed, and some certain tissues that are present in the mucosa called mucosa associated lymphoid tissue. I'll talk about them as well. So they're present. So lymph nodes, as you can see, they're literally present all over. I think the adult human body has like more than 400 lymph nodes. Um, The spleen is an organ in your peritoneal cavity that you can see over here. The thymus is this sort of butterfly-shaped organ up here, sort of at the top part of your chest cavity. And then in all your mucosal layers, so like in your nose, mouth, in your gut, and all these different mucous membranes, you have lymphoid tissues and sentinel cells. Then obviously your bone marrow in adults in the pelvis and so on. And your skin, of course, mucosa in the skin contains um, a lot of lymphoid tissues that lymphoid cells that are uh, actively moving around, trying to make sure that there's no harm. Um, yeah. So as you guys would have studied, I'm sure, in some varying detail, that there are some um, two different branches of the immune system, right? So there's the innate immune system, and the adaptive immune system. And so the job of the innate immunity is to provide an early line of defense against microbes, pathogens, anything. So there's a lot of cellular and biochemical defense mechanisms that are put in place with the help of innate immunity, where even before infection, they're just ready to act very, very fast against infections. As you can see, innate immunity, within 12 hours, you have a full-blown attack. Like, say you have a scratch on your skin, right? And some bacteria or something are trying to enter over there. What happens? Aside from healing, right, which is, sort of a warring thing, but you have healing of that cut, but also if there were some bugs that were trying to enter, then you have your white blood cells, your neutrophils, and your macrophages reaching there and making sure that they're cleaning up anything that is there that could be harmful that's trying to enter the body. So this happens, and you'll have that part inflamed, and you'll have like some reaction and inflammation going on in that place where you have a cut or something. Within 12 hours, you'll have a full-blown innate immune response. Whereas adaptive immunity, right, this effect takes longer. It can take up to about a week. So before I move to adaptive immunity, your innate immunity that consists of physical and chemical barriers like your epithelia. You have antimicrobial chemicals that are secreted. For instance, lysozymes are antimicrobial. That are there in your sweat, and is there in your saliva, and is there in your tears. Phagocytic cells, like I mentioned, neutrophils, macrophages. These are very important phagocytic cells, meaning that they can take up bacteria or pathogens or debris from cells and clear them out. And that helps to prevent tissue damage and infection. Then you have dendritic cells. These are professional antigen presenting cells, meaning that if there is some foreign cell or like a cancer cell or maybe a bacteria or some infected cell, they can take a little chunk of that cell essentially, which is foreign, and chop it up into little pieces and present it to T cells. So that's called antigen presentation. So dendritic cells and even macrophages, they can all do that. And NK cells, very important mediators. They act a lot like T cells, but they are not antigen specific. So they're not specifically targeting one particular protein, but they exhibit cytotoxicity like T cells. And then you have some innate lymphoid cells. You also have blood proteins like the complement system, for instance. And these are all what make up the innate immunity. So basically something that acts fast and does not have memory, right? Whereas adaptive immunity, which is your specific or acquired immunity, right? This recognizes and reacts against a large number of pathogenic substrates. And one of the defining characteristics of adaptive immunity is that it's able to distinguish different substances. Basically, it has antigen specificity, right? So that means that, say, suppose you get chicken pox as a kid, there's a particular set of proteins that are specific to chickenpox that now your adaptive immunity can recognize. So you won't get it again, ideally. Some people still do, but mostly you will not get it again because your adaptive immunity was trained and has memory to remember that array of proteins that it can associate with chickenpox. And so if that disease were to try to manifest in your body once again, your adaptive immunity would act so fast to get rid of it that you would never see any symptoms. So that's what it is. It has antigen specificity and it can react very vigorously upon a repeated exposure. So it has memory. And this uniqueness is what gives adaptive immunity this essential property. And some of the main effectors of adaptive immunity are your lymphocytes, like your B lymphocytes or B cells that differentiate into plasma cells when they're stimulated and secrete antibodies. And then you have your T lymphocytes or T cells that can be of many different types, but essentially they meet us if they come across a certain antigen that they're reactive against with the help of their receptor, then they proliferate into clones of themselves and then they attack it and try to get rid of it and then have memory so that if it comes back again, they'll react again. So that's your innate and adaptive immunity. So now coming back from the generalized principles of the immune system, and we'll talk about now important components of the immune system with respect to the immune organs. So as I mentioned, there are primary immune organs and secondary immune organs. So bone marrow is a primary one. So it's this, as you guys all know what bone marrow is, I'm sure, right? So it's this sponge-like tissue that's found in your bones. And as a kid, you would have red bone marrow in a lot more bones, but in adulthood, uh, only a few of our bones actually have red bone marrow. So that's the ribs, your breastbone, the pelvis. Those are the main regions where you have red bone marrow, which is where a lot of hematopoiesis takes place. And that is the process of generation of new blood cells, right? So here you can see this is an example of a bone, probably of someone younger, considering this doesn't look like the breastbone, ribs, or pelvis. So in this bone, which looks kind of like a femur, you have these different regions, right? The epiphysis, the metaphysis is the connecting region, and then the diaphysis is the long shaft. So your red bone marrow is typically in this region of the epiphysis, and that's where your hematopoietic stem cells exist, right? So every type of cell in our body, differentiated cell, arises from a certain type of stem cell. So for blood cells, you have hematopoietic stem cells or HSCs, right? For skin cells, you will have some skin stem cells for or epithelial-like stem cells, right? So you have different types of um, pluripotent stem cells that are available in different tissues that give rise to and help with the wound healing or help with cellular turnover in those organs. So in the bone marrow, you have hematopoietic stem cells, and they can give rise to your red blood cells, your white blood cells, and your platelets. So if you look a little deeper into that, you start with your hematopoietic stem cell, right? And you get these things after that. It can differentiate one step further into something that's not completely pluripotent. Instead, it has certain set number of different cell types it can differentiate into. So this intermediate phase is called a progenitor cell. So you can have myeloid progenitor cells, which largely give rise to your innate cells, innate immune cells. And then you have your lymphoid progenitor cells, which uh, largely give rise to your adaptive immune cells like your T cell and B cell, though NK cells are not technically considered part of the adaptive immune system, they still come from the lymphoid progenitor. So yeah, myeloid progenitor cells give rise to megakaryocytes, which then break into pieces and become platelets. Then you have eosinophils and basophils and neutrophils. These guys are your granulocytes, right? And they're really important when you get a complete blood count done, right? Your CBCs, you always get the numbers for neutrophils, basophils, and eosinophils, and neutrophils are the highest, right? Then you have monocytes, which go into tissues and differentiate into macrophages. They can also become dendritic cells, though dendritic cells have different, not just from monocyte lineage, there are other ways. Then you have your erythrocytes, which are your red blood cells. And then your lymphoid progenitor cells, they give rise to T cells and B cells. B cells can then differentiate into plasma cells that secrete antibodies. Okay. So these are your essential sort of cellular components, and they are all one way or another engendered in the bone marrow. Okay, so they all sort of arise from the bone marrow. So as I mentioned, bone marrow architecture changes, right, with age. So this is quite a lot of text. I apologize for that. But basically, this shows different bone marrow biopsies of people of different ages. So the first one, this is A, over here, the top left one. This is bone marrow from a 5-year-old child, and it's more than 90% of it has cellular components, right? And there's just a little bit of adipose tissue. So adipose is basically the fat tissue, and those are these little circles, the white circles. Then the B image, that's bone marrow biopsy from a 60-year-old adult, so quite a lot older. And this has about 50% hematopoietic elements and 50% mixture of this adipose tissue, which is the white circles. Then another interesting thing is a post-chemotherapy marrow. So you know what chemotherapy is, right? I'm sure. So chemotherapy is basically a bunch of drugs that are flooded through the body of a person who has cancer. And this essentially targets, well, a majority of them target act rapidly dividing cells and get rid of cancer in that way. But unfortunately, another thing that happens is that it gets rid of a lot of stem cells and it gets rid of a lot of rapidly dividing populations that are not cancerous. And it can actually, chemotherapy can make people very, very immunosuppressed. If you have someone in your family who's suffered from cancer, you would know that, people become very weak. They have to wear masks. They have to make sure that they don't go into, public spaces that could have a lot of infections roaming around, because you become really immunocompromised, right? You have a very weak immune system after chemotherapy. And this shows even at the level of the bone marrow. You can see that there are these large sort of gaps. Essentially, these are called sinusoids. So you have these dilated sinusoids, essentially these big gaps, and they have the cellular, the number of cells is decreased a lot. This is a little magnified, so it looks even like there's even fewer cells, but in general, the number of cells decreases a lot. So I just wanted to sort of show you these three different aspects of and the way bone marrow can look in young, old, and post-chemotherapy patients, right? Cool. So that was about the bone marrow, one of the essential places of hematopoiesis. Now, when you look at T cells, after they have been created, they go to this organ called the thymus. This is like a gland-like organ, and the T cells go over here to get mature. And I'll explain what happens in just a little bit. So this thymus is this sort of butterfly-shaped structure. It's right above the heart and lungs. It's right above this chest cavity region, and a little below the neck, right? And this is sort of what the histological section looks like. An interesting thing about the thymus is that it actually reaches full maturity in kids, in children. And that's where at you have, so this left one, A, is from a child. It's a thymus section from a child. And this shows like most of it is proper lymphoid tissue, right? 80% or more is lymphoid tissue. But over time, when a person becomes an adult, actually there's this process called thymic involution, where the thymus just sort of shrinks and also becomes mostly fatty tissue, which is what you can see over here. So all of these, there are still some functional units, so it's not like there's nothing happening, but there's a lot less of function in this thymus after about 20-21 years of age. So yeah, and so what is really happening in the thymus? So the T cells, they are, you know, they are at a sort of not completely developed stage when they leave the bone marrow. Okay, so they've been produced in the bone marrow, that's great, but they need to become mature. And for that, they go to the thymus. Now, as I mentioned before, you don't want immune cells not reacting against threats, and you don't want them reacting against our normal body cells. So you don't want them to underreact and you don't want them to overreact. So how do they know this? They are actually undergoing a training. So they go to the thymus and they are trained, and in fact 90% of T cells that reach the thymus die because they cannot pass this training, and only around 10% of cells leave the thymus fully trained, ready to react against the correct signals and not show autoreactivity. So this process is called positive and negative selection, right? And T cells undergo that at the level of the thymus. So you have an immature thymocyte that goes through some positive and negative selection. Basically, it's tested that it's reacting against the correct things, the correct threats, which are presented to it. So if it reacts, good, but if it reacts too much, not good. And it should not react against self molecules. So if it does, then also those cells will die. And then after that, they get some specializations, and then they leave the thymus, and then they are ready to go out into the body and look for threats. So at the level of the thymus, this is what's going on. Okay. So that's sort of, now that you know about more about the bone marrow and the thymus, this is the immune cell circulation that's occurring. You have hematopoietic stem cells that initially in the fetal level, they originate in the yolk sac. Then this hematopoiesis takes place in the fetal liver, and then eventually this happens in the bone marrow. And then these cells migrate through the blood to the thymus. This is the T cells, and then they differentiate. They don't differentiate into T lymphocyte. That's not really written correctly. Basically, they mature into naive T cells, and yeah, that's how it happens. And then these mature T cells and B cells, and again, this is not necessarily mature. I think this writing I'll change, is not fully accurate. Basically, these are naive cells that circulate in the blood, and then once they come across some threat, that's when they mature. And then they move through the lymph node, and eventually if they find the threat, maybe if there's a tumor growing in someone's leg and they find it, then they'll go and attack that tumor. So now I mentioned lymph nodes, and we have studied about lymph nodes earlier in earlier classes, so I can just go through it with you guys. So what is a lymph node, right? So lymph node is this kidney shaped organ, right? We have about 450 of them in an adult. And it's a very essential organ in the lymphatic system, and a very important site for the adaptive immune system specifically. So there's a lot of different lymph nodes linked through the body via these lymphatic vessels, and they are major sites where lymphocytes such as B and T cells are present. They filter through them, and they come across threats in them. So they detect whether there is some disease or some infected cell in the body somewhere else, not in a lymph node, somewhere else, that they need to react against, that they need to tackle. So those threats in the form of foreign antigens, foreign proteins, right? Those are presented to T and B cells at the level of the lymph node. So lymph nodes are basically like filters where B and T cells can encounter antigens or epitopes presented by antigen presenting cells. Okay, so APCs. And APCs essentially, suppose someone has some infected region in their body and some bacteria is infecting it. So these antigen presenting cells that are patrolling around in the tissues, they reach that site and they take little bites of the infected region, and they present, um, they also have receptors and stuff, and they present these, they move to the lymph nodes and they present these little bites of protein to the B and T cells, where they get primed. Essentially, they have a receptor and they bind with it and they get primed, so they know that there is this threat. Then they, make many copies of themselves, and then they go based on a lot of cytokine and chemokine signatures. So the lymph node architecture itself, it's enclosed in this fibrous capsule that you can see, and it's made of an outer cortex or cortical region and an inner medullary region or medulla. This hilum over here, this is an indent on the concave surface of the lymph node, and this is where your lymphatic vesicles are leaving and blood vessels are entering and leaving. Right, so the outer cortex has these, you can see these sort of blue blobs, right? So these are essentially groups of, mostly these B cells are inactivated. However, so they're sitting in these things called follicles. But when there is some kind of antigen that they're reacting against and they have been activated, then they form these things called germinal centers. And then in the germinal centers, they become activated, they proliferate, and then they become plasma cells and produce antibody. And there are these regions between the paracortex, which is where the T cells are, and the germinal centers, where T cells and B cells can interact with one another. And this is an example that I found in this paper. It's a really nice paper looking at essentially a lymph node. So you don't worry about all this stuff, but basically it is a live mouse that has fluorescent proteins that are marking these things. So XCR1 Venus is marking an antigen presenting cell. GFP OT1, this is basically marking T cells that are specific to a particular protein presented by the green cells. And TT tomato polyclonal CD8s, these are basically T cells that are not reacting against any specific. So they're just T cells that have not found a specific protein to react against. So they're just roaming around and looking for a threat, whereas the blue ones are the guys that have already located a threat. And this is inside a live mouse, so it's using a technique called intravital imaging, where they've imaged a certain organ or certain tissues inside a live mouse. Because if you take this organ out, the organ will start dying and it will not survive for more than 30 minutes, and that too on ice. So this is inside the live mouse using a very interesting and a very essential microscopy technique called two-photon excitation fluorescence microscopy. Essentially, it allows us to look at fluorescence in thick samples, which is what you need to get through a mouse. So they've used this technique, and so now I'll just play the video for you guys so you can see this. This is unimmunized. They've given some protein that essentially makes more of these blue cells to appear. So now you can see more of the blue cells, right? So this is 12 hours after. Poly-IC is just an adjuvant. Now you can see a lot more of these blue cells, big clusters of these blue cells. That's because there's been more time since, right? 46 hours. Look at how many blue cells are there. These are the blue cells that are actually going to react against um, this particular foreign protein. Okay, so ova is the foreign protein, and OT1 cells are the ones that are going to attack any cells that have presented this foreign protein. So over time, more and more of these cells are produced because they are making many more copies of themselves. So they have imaged this mouse for 46 hours, and this is what they see. So it's a very nice paper. If you guys want to look at more of the videos, you can check out this paper. Right. So that was about the lymph node. Um, Now we can look at another important organ, which is the spleen. Spleen is, you guys have all heard about the spleen. You can see it over here. It's in the peritoneal cavity. It's found in the upper left side of the abdomen. So if you're looking down at a spleen in a sort of dissected organism or human, it would be right for you. But if someone is lying down, it's on their upper left in the abdomen. It's beneath the diaphragm. It's behind these intestinal folds and behind the stomach. It's sort of back there. And it has many different jobs, of course. The major ones are that it stores immune cells. So you have a lot of T and B cells present in the spleen as well. You have germinal centers of B cells in the spleen, and T cells that come across antigens and interact with the B cells as well in the spleen. And if needed, these cells can move out of the spleen and travel via the bloodstream to different parts of the body. And you also have phagocytes in the spleen that help to, so phagocytes meaning you have like macrophages and stuff, tissue resident macrophages in the spleen as well, that help to clear out any sort of um, germ or sort of particular debris that might be cluttering up and dirtying the blood. This is also, you guys have heard, I'm sure, in school, the spleen is the graveyard of RBCs. So it's very true. It breaks down red blood cells. Those that are, no longer, so red blood cells only live for about 7 or so days, right? So after that, they're broken down in the spleen. Then it stores and breaks down platelets as well. So thrombocytes, they're also broken down by the spleen. And interestingly, spleen tissue is quite soft, and it can get torn or smashed very easily. And so if someone has an accident, people get splenic injuries in the peritoneum more than other parts of their other organs. And so often if someone has profuse bleeding in the spleen, instead of trying to repair it, people just remove it in surgery. So they do splenectomies, which is to remove the spleen completely. And in fact, other immune system organs can compensate for its job and they make up for it. It's like when people, have their tonsils removed or they have their gallbladder removed, right? The body compensates. Or you have one kidney removed. It's not exactly the same thing, but essentially the body finds a way to cope and we can survive without a spleen, although there are some lifestyle restrictions. The spleen is interesting. It has this white pulp and regions, and like I won't go into too much detail because it's just too much stuff, but it has white and red pulp, and those regions define what kind of immune cells are there, and you can read about it more if you like. Then we come to tonsils. You guys have all, I'm sure, heard of tonsils. Some people have swollen tonsils, or they say they have swollen adenoids and they have to get them removed. Nowadays, I think tonsil surgery is a lot less common. But in older times, I do remember people saying, "Oh yeah, my tonsils were removed," and like I think now they have ways to like not do that. So I think people still get tonsillectomies every now and then. So yeah, so there's different types of tonsils. You have a pharyngeal tonsil, tubal tonsil, a palatine tonsil, and lingual tonsil. So these tonsils all are in this kind of a ring of tonsil tissue, and it's called Waldeyer's ring, and it's around the opening of the mouth, of the throat, from the mouth and nose. Um, there's also lymphoid tissue that's on the side of the throat, that is not a tonsil, but if someone has their tonsils removed, their palatine tonsils removed, this lymphoid tissue can take over for that. So again, I'm saying like the body has a way to compensate for the loss of certain parts, not everything, but to some degree, the body can keep up. And yeah, tonsillitis is the inflammation of the palatine tonsils. People get it sometimes if they have a really, really bad cold or they have some kind of infection. It shows up in the form of like your tongue gets like white, like dots, and there's like halitosis, which is basically bad smell, and in the mouth, and pyrexia, which is fever. Um, yeah, so bacterial infections can cause tonsillitis, and typically antibiotic treatment and whatever the basic stuff would get rid of it. But if someone has persistent problems, then removal may be necessary. Yeah, and this is sort of what the palatine tonsils histological slide looks like. And moving to the final type of immune related tissue. So this is called, I don't know if many of you have heard of this before, but it's MALT, which is mucosa associated lymphoid tissue. So this is a very important component of the immune system. It's all over the body, and it consists of lymphoid structures that are distributed throughout mucosal surfaces. I was mentioning earlier, right, that mucosal surfaces like the gut, the nose, the bronchus, the skin, they all have certain lymphoid tissue. So that's basically MALT. And they are very important for acting as a sentry in those regions to make sure that harmful pathogens don't enter and we maintain tolerance to things that are harmless. So, have you guys heard of? I'm sure you've heard of gut microbiota, right? So that's just like millions and millions of bacteria that hang out in our gut, in our intestines. And so now how does the body know? And now bacteria is there in our body, but if some bad bacteria were to enter, the body knows who to react against, right? So why isn't our body reacting against all of the bacteria that exist in our gut, right? And that help with our digestion, help with maintaining, a good gut health. And in fact, without them, you can get dysbiosis, which is an imbalance of your gut microbiota. So why isn't our immune system freaking out and attacking them? It's because we have peripheral tolerance. We have this tolerance mechanism, essentially a way of these immune cells to not attack antigens related to those related to intestinal normal homeostatic bacteria like lactobacillus, for instance. Our body is not reacting against it because of where the lactobacillus is and the tolerance mechanisms in place to protect against excessive reaction against anything like that. So yeah, we have different types of MALT. We have different types of mucosa-associated lymphoid tissue. In the gut, you have, it's called gut-associated lymphoid tissue. This includes Peyer's patches, which you may have heard of, and some lymphoid follicles that are there in the intestine. Bronchus-associated lymphoid tissue that's present in the respiratory tract, and it defends against inhaled antigens. Then you have nasal associated lymphoid tissue, which is part of the upper respiratory system, and that's where it helps with immune defense right at the sort of opening. And then you have skin associated lymphoid tissue that is in the skin and comprised of Langerhans cells, dermal dendritic cells, skin resident T cells, and many other cells, gamma delta T cells, and so on. So there are all these types of different populations, and they're really spread out in very strategic ways to make sure that the right cell type is present in the right tissue to protect against different types of pathogens, because everyone can't be doing everything at all at once, right? So this sort of handing out of and distribution of roles is very well sort of, it's not very well understood in the immune system, but it is a very beautiful, beautifully planned system in a functional immune system. Right, so that was about the different immune system components, and I put together a few questions and answers, simple ones, just to recap what we have looked at today. And we can go through those, and then we'll be done with today's class. So, does anyone have any questions about this or any other topic that you want to discuss beforehand, before we go into the questions? Anything you're confused about? Anything you want to understand better, or something you want to know about the immune system? Everyone's all good? Okay, then I suppose I can move along. Um, right. So, I've got a few simple questions for you, just to know, just to recap. So you guys can send the answer that you think is correct in the chat box and we can discuss. So which is the primary site of hematopoiesis in adults? Do you guys know? Anyone want to try? You can send the answer in the chat. If you're not sure also, you can send. It's fine. Yeah. Okay, awesome. So you guys are right. It is A option. It's the bone marrow. So that's where in the red bone marrow, very specifically, is where in the epiphysis region of the bone is where you will find hematopoiesis going on a lot. And in adults specifically, because in like fetus, that's not where it happens, right? So it starts at the yolk sac, then in the fetal liver, and then when a baby is born, eventually this hematopoiesis moves to the bone marrow. And the process of that is very interesting. So yeah. Okay, here's another question. So what process is occurring in the thymus? Do others want to try? Yeah, great. So I'm seeing all of you have got the right answer, which is T cell maturation, or in fact, it could be more like T cell selection, because essentially the T cells, which are at a sort of double negative unselected stage, they go through a selection process, right? And then they, at the end, they are still naive in the sense they have not experienced antigens, they have not experienced like immune activation. So maybe T cell maturation is not the most accurate thing for me to put as an option here. Instead, it should be T cell selection. So yeah, they undergo T cell selection or a selection process, refinement, one would say. Okay, let's try another one. So, which structure in the lymph node is primarily associated with B cell activation? Do others want to try? Okay, so I've got someone saying A, someone saying C. Anyone want to try another option? Yeah, so the correct answer here is C, which is the germinal center. So the paracortex, which is option A, that is actually where T cells are present majorly. And the germinal center, which is present a little inner to that, which is in those B cell follicles essentially. Um, so between the paracortex and the medulla, you have the follicles within which there are German centers. So when B cells are activated, so B cells basically they sort of mill towards the side of the paracortex within the follicles, and they interact with T cells. And when they're interacting with T cells, sometimes they receive a stimulus from T cells, right? T cells that have been activated themselves, and they then they get activated from that um, from that stimulus, and then associated with B cell activation, they form these germinal centers, right? So there are these structures that actually appear when B cells are multiplying, okay, in response to some stimulus or in response to some specific antigenic signal. Um, and those germinal centers are what you see in the lymph node, and they are associated with B cell activation. So I hope that's clear now. Okay, let's do a few more questions. So what is the main function of the spleen? Do others want to try? Anyway, in interest of time, I'll just go ahead. So it is, you're right, it's C option, which is that it filters blood and removes old RBCs, removes old platelets as well. So that's why it's called the graveyard, right, of RBCs. It's because old RBCs are um, sort of broken down by the spleen, and then their individual components, heme and all of that, it's removed and then recycled. So spleen does this. It's like a factory that disassembles RBCs, takes what's important, throws out what's not important, and then recycles whatever it can. All right, let's do another question. So which of the following is a component of MALT, or mucosa associated lymphoid tissue? So I mentioned these. Okay, getting some answers. That's good to see. Yeah, so good. You guys are right. It is B option, which is Peyer's patches. So you may have heard of Peyer's patches before, right? So they're specialized lymphoid structures that are there in the small intestine. Mostly it's there in the ileum region of the small intestine. And very important, they're called GALT, G-A-L-T, gut-associated lymphoid tissue, which is one type of MALT. Um, yeah, so typically they're sort of these aggregated like nodules of like lymphoid nodules, which contain B cell follicles, T cell zones, dendritic cells, macrophages. Everything is there. It's like a mini lymph node, sort of peppered across the gut. And Peyer's patches specifically contain these very important cells called M cells or microfold cells. And these are very important for taking up antigens from the gut lumen and then delivering it to immune cells that are there behind it. And interestingly, if you guys have heard of salmonella, salmonella actually makes its way through the M cells. So they get thwarted by salmonella, and we don't, there's a lot of research going into why that happens. So salmonella sort of manages to cross across M cells in the intestine and infect hosts, right? So it's generally Peyer's patches are very important for intestinal immune surveillance, which is basically the process by which immune cells are making sure that nothing evil is crossing into the body, right? No bacteria or viruses or infected cells or debris are able to get through. But of course, sometimes there are mechanisms that the pathogens employ to hoodwink the immune system and get through anyway. Yeah. So yeah, that's generally it about Peyer's patches. Right, let's do another question. So, which of the following best describes innate immunity? So, we talked about this, right? Two branches of immunity. Any idea, guys? Yeah, so you guys are right. It is B option, which is that it has very rapid response, but it's a non-specific response with no memory. So that's B option. The other descriptors are more specific to the adaptive immunity, right? Antigen specific, has memory, requires prior exposure. Well, I mean, not necessarily, not for adaptive. And yeah, so good, great. Let's do a few more. So which immune cell type is primarily responsible for antibody production? What do you guys think? Anyone? Okay, so maybe you guys got a little bit thrown off by this one, but basically the answer here is C option, which is plasma cells. So plasma cells are basically B cells that have activated and differentiated into this slightly different secretory type of plasma cell. So B cells have B cell receptors on their surfaces, and when they're activated, they actually are able to throw those receptors out in the form of antibodies, immunoglobulin based antibodies, right? You guys have heard of, IgB, sorry, IgA, IgG, IgD, IgE, right? So these are different types of antibodies that are secreted by B cells when they have differentiated into plasma cells upon activation because of some specific antigenic signal. Okay, so the correct option here is option C. I hope that is now well understood. All right, so which region of the lymph node is rich in T cells? I think I discussed this just a few minutes ago. What do you guys think? Anyone? So yeah, so the correct answer here is I can understand why you'd say cortex and you're very close. It's actually the paracortex. The paracortex, or it's known as the deep cortex, that's the region where T cells are there the most. The T cell rich region is the paracortex. So it's between the outer cortical area and the medulla. And that's where a lot of dendritic cells are there, which present antigen to T cells, and that's why T cells are also there. So, and another reason why there are a lot of T cells in that region is because there's this thing called a high endothelial venule, HEV, and these bring a lot of lymph in. So they are, sorry, a lot of blood in, and they are an entry point for naive T cells from the blood. And those HEVs are where they contact the lymph node at the region of the paracortex, which is why you see more T cells there. Yeah, so the cortex has your follicles and your B cells, your paracortex has your T cells, and your medulla has plasma cells, macrophages, and other stuff. Yeah. So, all right. So, which of the following is a function of tonsils? Yeah, you guys are absolutely right. It's option C, which is it generates immune responses against antigens. So the other ones are not accurate, right? Filtering blood is for the spleen. A site for T cell maturation is a thymus. Destroyed age platelets as the spleen again. But this is the right one because that's it. Tonsils has all of the sort of immune function related cells. It has phagocytes, it has T cells, B cells. So it can generate immune response against antigens, specifically those maybe that are um, much more peripheral, meaning like your maybe that come in with your food or while breathing, right? Okay, few more questions and we're done. So which of the following cells is part of the innate immune system? Any idea? Right. So in interest of time, I'll just tell the answer. The answer is monocytes. Monocytes are the component of innate immune system. Yeah, you're right. D option. So monocytes differentiate into macrophages. These are all part of the innate immune system. They are particularly specialized in taking up um, debris, dead cells, pieces of dead cells, bacteria, virus infected cells, and so on. So yeah, monocytes are the important component here. The other guys, B cells, plasma cells, memory T cells, they're all adaptive immune cells. Okay, where does negative selection of T cells primarily occur? So this is a bit more specialized, my bad, but yeah. So the correct answer here is well, I guess the main thing here is that it happens in the thymus. So positive selection typically happens in the thymic cortex and negative selection occurs in the thymic medulla. So the correct option here would be C, but I think for your purpose, the main thing is to know that selection is taking place in the thymus. All right, last question. Which of the following best distinguishes adaptive immunity from innate immunity? Yeah, yeah, absolutely right. It's option D, which is antigen specificity and memory. Yeah, great. So that's all I have for you guys today. I hope that's clear, right? That last question, that it's pretty clear, right? Antigen specificity and memory is a characteristic of adaptive immune cells, whereas innate attacks immediately and does not remember. Yeah, great. So I hope that was sort of useful to you guys. And these topics, I mean, some of it was shared with what you have already learned, and some of it was new or not covered in this in the syllabus of this course. So yeah, if you have any questions, let me know. We can discuss. Otherwise, I will sort of adjourn the class here. And there'll be, I think, one last class of this course where I'll continue with the recap of topics from the course, important concepts and stuff. I'll go over them with some more questions, some higher order maybe. And that'll be in the same timing as my normal TA class, which is on Tuesdays next week, from 7 to 9. Yeah. Okay. All right then. See you guys next class, in the last class. Yeah. Bye everyone.